the final video in our series looking at the composition of blood, we're going to be taking another look at the blood group marker proteins, which are a group of identification markers that are found on the surface of your red blood cells, and looking at how, even though they are naturally occurring within the human body, they function identically to antigens that are found on the surface of pathogens like bacteria and viruses. So as a review, your blood group markers, at least in what we call the ABO marker family, have three different varieties, the simplest or default form being the O marker, which indicates whether a person has blood type O. This is the default form of the blood group markers, in that the other two markers, what we call the A antigen and the B antigen, are structurally identical to the O antigen, just with two specific modifications that are present. If the O antigen is modified in one way, this will give a person blood type A rather than blood type O, and if it is modified in a structurally distinct way from A, this would give a person B-type blood. Now, blood type is probably the best example of what we call multiple allele inheritance, as well as codominance, in that if a person inherits the A antigen from one parent and the B antigen from another parent, their red blood cells will actually contain both blood group markers and therefore end up with type AB blood rather than A or B on its own. Now, a third example of a marker protein which is inherited independently of A, B, and O, meaning that it is controlled by a separate gene, is another marker protein that we call the Rh factor. Now, we can see this red blood cell here, if we represent the Rh factor by these little black markers on the surface, we can see that this red blood cell is what we call Rh positive, because the Rh factor is present, whereas on this red blood cell we see no Rh factor here, and therefore we would label this red blood cell as Rh negative. And for blood typing identification purposes, it's customary to list both the A, B, or O blood type first, and then whether the Rh factor is present second. So blood type O positive means that it has the O antigen of A, B, and O, and the Rh factor is present on the surface, whereas with blood type O negative, the Rh factor is nowhere to be found. Before we begin looking at the roles of antibodies, there's one topic that we can cover here, which is one possible reason why A, B, and O blood type markers evolved in the first place, and that is as a method of protection against blood contamination. Now, there are many examples of pathogens that actually spread using contaminated blood. Probably the most commonly occurring is a parasite called malaria, which many people will understand is actually spread when mosquitoes suck the blood of someone who has malaria, because the malaria parasite actually uses your red blood cells to hide from the immune system, and if a mosquito transfers contaminated blood with malaria from one person to another, it makes it very difficult for your immune system to combat this parasite. However, if your circulatory system and immune system have a way to distinguish your blood as being distinct from somebody else's blood, that makes it easier to recognize and therefore destroy blood that did not originate in your body in the event that it contains a parasite such as malaria. And the way that your immune system deals with this is it recognizes that A, B, O, and Rh are all examples of antigens, which means that your B cells will produce antibodies that can target these antigens. Now, the way to determine what kind of antibodies your B cells will produce is to look for all of the antigens that are not found in your blood. And this is probably easiest if we take a look at the blood types individually. For example, if your blood type is A positive, that means that your red blood cells have the A antigen and the Rh antigen, meaning that the B antigen is the only one that is not present. And since it's probably not a good idea for your B cells to make antibodies that will attack your own blood, this is why a person with blood type A positive will make antibodies that target the B antigen specifically, because this is the only antigen that is not found in your blood. This is not true about A negative blood, because A negative blood has the A antigen but no Rh factor, and therefore they would produce antibodies that would target not just the B antigen but also produce antibodies that would attack the Rh factor. Now, blood type B negative and B positive, respectively, are easy to predict as well, because B positive has the B antigen and the Rh factor, and therefore the only antibodies that they would produce would be antibodies that would attack the A antigen. But with B negative blood that only has the B antigen here, they would make not only antibodies that would attack the A antigen, but also antibodies that would attack the Rh factor as well. Now, AB and O type blood are interesting because they are diametric opposites of each other in terms of how antibodies are produced. So because AB positive blood has A, B, and Rh antigens found on the surface of their red blood cells, that is all three of the distinct antigens that can be targeted by antibodies, and so people with AB positive blood do not produce antibodies that attack any of these, because that would involve destroying their own blood, which is why people with AB positive blood are recognized as what we call the universal receivers, meaning that people with any other blood type can give to a person who is AB positive. AB negative is close to this, but since they only have A and B and not the Rh factor, that means that the only antibodies they would produce would be antibodies that attack the Rh factor. If we go to the extreme O negative blood has none of the three critical antigens, A, B, or Rh, and therefore it would produce antibodies that attack the A antigen, B antigen, and the Rh antigen, which means that O negative people, or people with O negative blood, can only receive blood from others that have O negative, because they produce antibodies that would destroy any other blood type. Whereas O positive blood actually has the Rh factor, and therefore their B cells would only produce antibodies that would attack A and B antigens respectively. Now, when we say attack, we can take a look at what the consequences are of antibodies actually attacking red blood cells that are not found in your body. So if a mismatched blood type, meaning a blood type that is not suited towards your own, or a blood type that can be targeted by antibodies, that's exactly what we would see happen. So, for example, if our blood type is A, that means that our B cells would produce anti-B antibodies, and if we received the wrong blood type, if we received B-type blood here, the antibodies would bind to the wrong antigens that we see here and cause red blood cells to agglutinate like this. And this phenomenon is what we call hemagglutination, which literally means that your red blood cells would stick together like this, because one antibody can immobilize multiple red blood cells like this, which makes them harder to move throughout the body and easier for your white blood cells, specifically your phagocytes, to track down and then absorb and destroy, potentially killing any parasite inside of them. The problem is, and the main reason why receiving a mismatched blood type when you are supposed to receive your blood type, is because of the negative consequence that can happen with hemagglutination. If hemagglutination leads to a large number of red blood cells accumulating and sticking together like this can actually cut off the circulation of blood in some of the major blood vessels in your body and lead to blood clots that eventually prevent oxygen from being able to reach tissues. And if this happens in your heart, that's what we call a heart attack. And if this happens inside of your brain, that's what we call a stroke. So it's very important whenever you receive blood to make sure that you know what your blood type is in order to determine which type of antibodies are produced and how you can avoid the process of hemagglutination by receiving the wrong blood type. This concludes our series looking at the composition of blood. Before you move on to the next video series that will look at the heart and the mechanics of the heart, take a look at the case study below and see if you can use your knowledge of blood in order to explain how blood loss is significantly detrimental to the body by looking at how blood loss would affect the major organs of the body, and then take a look at the practice questions that follow this and see if you can test your knowledge of the importance of the different components of blood.